hola como compañeras y profesores y todo. Hoy en el seminario tenemos dos, Silva y Liliana Bustos. Los dos son compañeras de doctorado de Neurociencia en la Universidad de Valparaíso. Nuestra primera expositora va a ser Valeria Silva. Valeria es biotecnóloga de formación. Ella hizo su pregrado en biotecnología y después hizo un magíster también en biotecnología en la Universidad Andrés Bello de Santiago. Luego ella se quiso cambiar un poco de ciudad y vino a Valparaíso, empezando a trabajar en el laboratorio de Dr. John Ewer como asistente de investigación y durante 3 años. Y porque a ella le gustan las moscas y las abejas también. En 2018 ella ingresó al programa de doctorado en ciencia, mención neurociencia también en la misma universidad de Valparaíso, continuando trabajando con John. Y hoy Vale nos va a presentar algunos resultados de sus tesis de doctorado titulada Role of Eclosion Hormone During Ecdysis Behavior in Drosophila melanogaster. Ok, Valeria, puedes compartir pantalla y empezar tu presentación.
Here. So can you see it perfectly? Eugi, can you see
Estamos y te vemos.
Yeah. Yes. Yeah.
Sí, está bien.
So thank you, Jim, for this nice presentation. I'm going to talk about the role of eclosion hormone in Drosophila melanogaster ecdysis. I know this is a different topic in this Center of Neuroscience, so I always like to introduce my research mentioning or showing this graph that shows all the species named on Earth, and insect and arthropod are today the most diverse and the most ecological dominant animal here in this planet. So one of the reasons of why this possible reason that they have this success here is because they have a small size, big offsprings. They also have a short life cycles, and they do have exoskeleton. This exoskeleton is a strong structure that avoid them to desiccation, also protect them from injuries and allow them to adapt to climate change. So the problem of this exoskeleton, well, it's not really a problem, but in order to grow, the animal must eliminate this structure in order to grow, right? So this is an example of the one beautiful insect called monarch butterfly. And you can see here, there is an egg, all the larvae stage, then is a pupae, and finally is a adult butterfly. So in every single stage, this animal must to eliminate the old cuticle and synthesize a new one, right? So this is the molting period, and this culminate with a super stereotype movement of muscle that allow animal to release the old cuticle that is called ecdysis behavior. And this is the main topic of my thesis. So in the lab, for obvious reason, we don't work with monarch butterfly. We work with Drosophila melanogaster, which is a super cool model. Anyway, we know all the genome of this animal, and also have many genetic tools to study this animal. And it's a nice model for ecdysis behavior because they perform ecdysis are at the larvae stages, also at the pupae, and then eclose the adults. And this is conserved across the insect. So here there are 3 videos of different ecdysis behavior of the Drosophila melanogaster. This is the larvae ecdysis, this is the pupae ecdysis, this is the adult ecdysis, also called eclosion when the adult emerge. This is a super stereotyped movement that allow animals to release the old cuticle. I hope you can see the videos. So this is the larvae performing the ecdysis process, so there is going to be release. also the pupae, and this start the movement, and you can see here there is the detach of the old cuticle, and eventually the head emerge, and finally the adult. This is a bit longer because the adult has to dry itself, and eventually, I will move further this, they inflate the head and they open the operculus, and then they close super fast. So even though these three stages look super different, they are, the ecdysis is conservative and is controlled by the same amount of neurons and for the same hormones and the same neuropeptides. I did this draw in order to simplify the ecdysis and say to you that there are two main neuropeptides that control behavior. One of them is synthesized in the brain, which is the red here, larvae in the pupae, and this is the brain of the adult. And the other is expressed in the trachea. This is the trachea, the respiratory system of the Drosophila, the insect in general. And here you can see, and in the brain, in these two neurons, you can see here in red, is expressed this neuropeptide eclosion hormone, and in the trachea is expressed this another peptide called ETH, ecdysis triggering hormone. So what do we know? Today we know that injection of any of these peptide, the eclosion hormone or the ecdysis triggering hormone, trigger the ecdysis behavior. I mean, if I inject this in an animal that is mature, I will promote the ecdysis. Also, we know there is a feedback between these two peptides. So the EH promotes the release of the ETH from the tracheas. And also we know if we do a genetic null of this peptide if you do a null, genetic null, so you have a larval lethality, so they die, this is a lethal phenotype, and we have the same phenotype if we eliminate the ETH. So I did this scheme trying to simplify what we know. So these are not only the two neuropeptides that control the behavior, there are many more. I'm going to simplify this, I'm not going to mention, but these are the beginners of the behavior. And it was published that ETH has its receptor and is characterized. We know exactly in which neurons and which cells express the receptor, what is their function, what is their temporal action, but we don't know the same about the eclosion hormone. So both are super important, but we only know the details about the ETH and not the EH, even though the EH was discovered before, it was discovered in the 1970s. So the main focus of my thesis here is to understand what is the role of the eclosion hormone. So this is a formal slide showing that the strategy that I'm going to use to characterize or to understand the eclosion hormone, what I'm going to do is characterize the receptor of this hormone, see where it's expressed and what the target cell does during ecdysis. So my objective one is identify as much as possible all the neurons and cells that express the receptor, and then is analyze the role of these cells and neurons during ecdysis. And finally, if we are lucky and the pandemic allowed me to do this experiment, I'm going to determine the timing of action, the neurons that express the receptor during ecdysis. So I mentioned the flies has these wonderful genetic tools. This is a super easy way to explain, but to identify the neurons, we use this binary system called USCAL4. I know I have mentioned this before in other presentation, but in brief, so we have an enhancer with a CAL4, which is a transcriptor factor that when it's together in a cell with this sequence, so promote the expression of any gene of interest. So in this case, we have a collaborator in the United States called Benjamin White, who did these flies. He did a fly, a GAL4 line that expressed the receptor. So have the EH receptor and a GAL4. So we cross with the GFP flies. So when they are together, all the neurons that have the receptor all the cells are going to be green. There are some variation of this technique. One is the split Cal4, that they divide literally the Cal4 into domains. So this allows us to see when two peptide or two protein are co-expressed in the same cell, they are going to reconstitute the Cal4, and we are going to see, for example, the GFP if we have our gene if our gene of interest is GFP. For example, if I want to see that EH receptor is colocalized, this symbol means that they colocalize with the ETH receptor, and I cross with GFP, the cells that express both of them are going to be green. And finally, we have this technique that is the Cal80, is a protein that blocks the Cal4. So then you can block the expression of the GFP in a group of cells, or the Cal4 in any place you want. So these three are nice strategies to work with genetics in flies. So the first objective was to see where the receptor is expressed, and as I mentioned, we have EHR with the GFP. So the receptor is expressed in a wide number of neurons and glia and cells in the brain on the central nervous system of the larvae. So this is the larvae, and this is the brain. This is the shape. So this is brain, and this is ventral nervous system. And here we have the adult brain and then adult ventral nervous system, and we see that the receptor is expressed in many places. So to see which are the identity of these cells, of these neurons, we start using everything we have in the lab. The first thing is the immunohistochemistry. So again, this is the pattern of all the neurons in the larvae. And one of the two peptide that we know that are involved in this behavior is eclosion hormone. So we have antibody of eclosion hormone, and then we can see in panel B, the same neurons that express the receptor, the synthesize the peptide also express the receptor. So we assume there is some positive feedback there or negative feedback, I don't know. And the CCP neurons that I just mentioned, also involved in the behavior and downstream of this peptide, express the receptor, which is super good because it was what we expect. So I did this in this stage, which is a larvae, a big larvae, almost PP, because it's easier, but I wanted to see if the same pattern are in the adult. So these are the adult flies. I didn't use immunohistochemistry here. I just put two drivers together. So in green is the EH receptor, and in magenta are the neurons that I was expecting to see. And again, I see the couple of neurons here that express EH has a receptor. So there's the colocalization and the CCAP as well. Using the split technique that I mentioned, I did a screening with all the split availables, and I find that there are so many subtypes that I can study that express the receptor. So they are target of my hormone of interest. I'm not going to go in detail of who are these neurons, but I wanted you to know that we know identity of subgroups, and we have this technique to study in the future. For another hand, we have find also that EH receptor is expressed in all the epithelium of the trachea. I told you before that there's a cell in the trachea that expresses one important peptide called ETH, which is in C here. So the cell that expresses ETH also expresses the receptor, so they have this feedback that we were expecting. But we also found that all the epithelial cells, how you can see in B or in this panel, which is super bright, all the cell has the receptor. So we don't know exactly why. There is another guy in the lab working in this. That could be something related with air absorption or air movement or water absorption. So we don't know, but it's nice. Additionally, we try to see where else the EH receptor is expressed, and there is expressed some in the cuticle. This is the larvae 1, 2, 3, the prepupae and the adult. So we know now, today we know there are some cells that has a receptor, and we are not going to focus this in my research, but it's good to know for future research of somebody. It's interesting because in the adult we see in the epidermis, but also we see in the proboscis, which is the mouth. of the adult. So it's nice to see. So the second objective of my thesis is analyze the role of the EH receptor neurons, so the target of the peptide. And to do this is my setup, so I need a microscope, a camera, and an agar dish where I can put my larvae and record it. Also, I can record pupae or adults. So I observe them in normal condition, and as well to analyze the role of this group of cells. The thing that I do is study the loss of function. To study this, I express the inward rectificator potassium channel, the 2X Chi-R2.1, that hyperpolarize the cells, so I inhibit the cells, so they're not responding anymore. I express this in all the cells that express a receptor or EH. And also I did the same experiment with Reaper, which is a apoptotic gene, inductor of the apoptosis. So this is what I see when I study larvae. You can see here, this is a success larvae, which is going to behave, and the ecdysis is the stereotypied movement that I can quantify, and eventually the larvae can be released. You're just going to see that they're going to release the teeth and eventually eliminate all the cortical there. And at the same time, this video is working, but they are not doing anything. So when I will just put a little faster, but they do random movement and they try to eliminate the cortical, but they don't. So they fail in the process, and this takes super long, maybe 2 hours against 20 minutes. This is a specific moment. The whole process is 30 minutes, but it happens in, I don't know, 5 minutes. And this is 2 hours video and nothing happened. So if we can quantify this, we can say that ablating or inactivating the EH receptor cells provoke larval lethality at the first ecdysis. So the first ecdysis they have to do, they all die. So this is how they die with the Chi-R2.1. So they get trapped, they cannot release it. And with Reaper, they also get trapped, but the trachea, because it's an apoptotic gene, the trachea start dying. So this is the quantification of the locomotion. This, and this are the genetic controls. And this is how they perform with Reaper. So they move a lot, they try to do ecdysis, but never end. And in the case of Chi-R2.1, they do atypical movements here for 10 minutes, and they spend like an hour or more trying to do ecdysis, and they partially release part of the teeth. So I consider it like ecdysis, but then if we see here in the quantification, neither the flies that express Reaper in all the EH receptor cells or Chi-R2.1 are able to complete the behavior. Their genetic control does. So this is another observation, like the timing of the tracheal collapse to the air filling, which also is a super important step of the process. They do not perform well, and they can never complete the air filling. They can never fill all the trachea, so they die, probably they die trapped or without oxygen. So this is a quick resume for this. So we did before, we published last year that if we express the same driver in the neurons that express the peptide, all the larvae with Reaper or Chi-R2.1 die during the L2 just after ecdysis. So they die in the super early stage in the larvae. But when we do with the same with the killing the receptor, they die during the ecdysis, which is a little more dramatic, but also there are more cells, so I was expecting to be more dramatic. So I cannot study with this driver in adults or pupae because they all die, but we have this another genetic tools called GAL80. So we use a temperature sensitive GAL80, which allow us to express the KIR2.1 or the Reaper at a specific point of the developmental, so we can study what is the effect only expressing this during the puppy ecdysis or the adult. So here you can see that if this is a temperature sensitive cal 80, so it doesn't work at 18, I mean, it's blocking the cal 4 at 18s, and when we change the flies to the 28 degrees Celsius, the cal 80 release the cal 4, so the drivers are going to be expressed. So this is the line time of how Drosophila is development at this temperature is at 18 take a lot of time, and then 28 is super faster. So we play with this shift of temperature to express or not our drivers. So to observe the pupae, I change here in the day 9 the temperature for the pupils, and if I want to study adults, I just on day 10 I collect the pupae and wait 6 days. I put in a new tube in order that they have all the same age or similar age, and here on day 16 I increase the temperature. What we observe, so if we express the KIR2.1 or the Reaper, this is a normal ecdysis. So this is the pupae, and the pupae do ecdysis like this. This is a normal one. So there's this pattern of movement that I can measure exactly. And this is how it works with the KIR2.1. So they just, I will put again because this is doing ecdysis pretty well. So this movement, and this is just, they don't do movement, just they attach the cuticle and go down. So the resolution is not pretty well. So I put this picture to see this is how a successful ecdysis work in pupae, and this is how they look when I use the KIR2.1 at this specific stage. So they detach, but they die in the other corner, or they just don't do the behavior, the stereotypic behavior. So in the adults we see something similar. So we here are going to see how the adult inflate the head right here. So the adult is going to make this movement of the ecdysis, and eventually is going to open the tuberculous here and release itself. This is the control. And in the case of these flies, I don't show a video because it's a 13 hours video. They do nothing in 13 hours. Actually, I record for 48 hours and they still do nothing. So they start inflating the head like this, and then keep inflating the head again. So inflate the head is essential because they inflate the head and then move open the tuberculous and go out, but they don't do it. So here's the quantification using the KIR2.1 in pupae. So this is the percentage of behavioral success and the genetic control here, and here is the with the KIR2.1, and they do not behave. They just has a failure there. And in this case, in B, we have the same in adults. They do not success, and using Reaper, I don't have yet the result of the pupae, but I do have with the adult, and around 20 to 30% they eclose. It's less dramatic than the KIR2.1, but they are different drivers, so we don't know exactly why one is more dramatic than the other, but still 75% of the pupae die. I mean the Farate adults, so the adult die, they do not eclose. So brief summary and then here, so I identify the neurons and cells that express the age receptor in Drosophila, which is my first objective of the thesis. And second, to understand the activity of this, I studied the loss of function of all the cells, ablating or inactivating all the cells that express the receptor. And if we don't have these cells functionally, they provoke ecdysis failure. I see this at larvae stage or at pupae and atelectasis. So what is next? Because I see like if I just turn down the system, it doesn't work. So I have to analyze the role of the subset. I mentioned before, I have this split gall line that allow me to study subsets. So I have to study subset of cells and see what is their implication during the ecdysis behavior. So I'm working on that. I try to express RNA in the specific cells, but the RNA is not strong enough, but so it's not really quantifiable. It's not clean the results. So last year it was published this super cool paper, which is in flies. We move one step in the future for the CRISPR process. So what they are doing now is we can use CRISPR to edit specific genes, right? But we are expressing with the GAL4. You can drive the CRISPR action and the guide RNA to a specific tissue. So for example, if I have a fly here in this column that has any GAL4 for any tissue and also the Cas9, and you express with your guide RNA to guide the mutation in any gene, and then you cross it, and you are going to have a progeny that can express the CRISPR and the guide RNA, for example, only in the wings. So you will have mutants only in the wings, or only in the gut, or only in the legs. So this is super helpful for me, because I want to do a tissue-specific mutation of the receptor to see the loss of function, right? So this is the gene of the EH receptor. It's has 4 isoform. It's not that big because it's the part that is going to be transcribed. And using the strategies that they mention in the paper, they say you have to design 2 is a pair to improve the CRISPR efficiency, a pair of guide RNA in the first exon that is going to be transcribed. And so they look each other, and if one guide RNA works or the other or both together, you will have more probability to have a specific mutation in your tissue specific. So when I was designing this, at the same time, a woman called Trivedi, she published the same, but she was using a guide RNA in the first exon and one in the last exon, looking at each other. So she said that with this technique, you can delete even genes with 17 or 18 kilobases, and this gene is only 5 kilobase. So I talked with both of them, and both were super enthusiastic about this. And they say, yeah, this is gonna work. Because science don't always work, I did 4 different pair of guide RNA. So I put in a plasmid, a specific plasmid for flies, and I cloned the competent cells, and we obtained colonies, and I sent this to Korea, I think, where they inject the flies, and they put this in a specific chromosome, and we got this fly. We have the guide RNA, and also have the Cas9, which also has an improvement to be less toxic in the tissue. So I have these 2 things in one fly, and now I'm in this step. I'm just 1 cross apart of the real result of my thesis. Sorry for that. And so I'm going to cross these flies with the tissue specific, with the EH receptor GAL4, with the in the different subset of cells. So the idea of this is to see how the behavior works, how the behavior is complete or not, turning out the receptor in a specific group of cells. So this is to be continued, and I hope it works. I hope so. The last objective of my thesis is to determine the timing of action of EH receptor neurons during the ecdysis. So probably most of you have seen this before. There's a guy before me who did wonderful videos of ex vivo real-time calcium imaging. So what I have to do is express with my EH receptor GAL4 this sensitive calcium zones in the cells, and then stimulate ex vivo, take off the brain, ex vivo with the peptide. So they did with the ETH. Now we have the EH. We have to prove it, but it was standardized with the ETH, and we have the spinning disk or to record this. And this is the video that we first observed. So these are all the EH receptor neurons, so all the neurons that are going to respond to my peptide. And they do have this, I want to put again. So they have this beautiful pattern of activity that is related with ecdysis. So the main goal is to be able to do this with the subset of cells, and that's all. So in conclusion, what I'm doing is characterizing the neurons that express the EH receptor, and that will help us to elucidate the role of the eclosion hormone and better understand this hormonal control of ecdysis. So here we have the model again. So we know the ETH is expressed in the EH neurons and also in many other neurons, and this is all known, and we are trying to see where the receptor is expressed. We already know it's expressed in the cell that synthesize the ETH. So this is a check. Oh. Also, we know many other neurons, so now we are in the step to see how exactly one group of neurons act first, then the other, and so on, and that's all. So I want to thank you, all of you, for listening to me. I want to thank also the lab of Peces y Moscas, which are great people. My mentor, John Ewer, who made me do all this science now. He allowed me to be in his lab and be happy there. And finally, to Dr. Benjamin White, who is is one of the god in genetics. And I just finished a 6-month internship just the last Friday. So today was my last day for quarantine. So I spent 6 months at NIH doing this experiment, which was really great. So that's all, and this is a publication of saying away that if you want to see. Oh.
Okay, gracias, Valeria. Y ahora vamos en las preguntas. Si en el público hay alguna persona que quiere hacer preguntas. Sí, Kate puede hacer la pregunta. Sí.
Nice to see you, Kate. You are mute.
Estás muteado.
Mucho tiempo. Felicitaciones. It was a really lovely presentation, especially without having done any practice. Anyways, could you remind me, you're inactivating the cells that express the ecdysone receptor. What is the actual mutants in the ecdysone receptor? What is the phenotype of a mutant in the gene in contrast to inactivating the cells that express the receptor?
Oh, they die. So we have the lethality is 90% in the. So we have two Gulfur line, but they die during the larvae stages.
Okay, so that's if it's a null mutant for the receptor.
Yeah.
And then if you look at the picture you showed, that is the co-localization of ecdysone and the ecdysone receptor, the cells in the brain.
Mm-hmm.
Can you knock out just those two cells?
Yeah. Yeah, the only two. Yeah, we did this. We just published this the last year, and we obtained that only the two of them.
The ones that co-localize ecdysone and the receptor. Yeah.
Yeah, we did that, and they also die during after ecdysis. So they passed the first ecdysis, but they die after.
Okay.
But no, they don't die during. So there are some that they complete the behavior, but also die. We don't know why.
So my question is, do you think that the lethality is only linked to those two cells and the rest of the cells are just kind of fine tuning?
Not really, because of the other, when I kill all of them, they die before.
Right, but you're killing all of them, but you're killing those two also.
Yeah, true.
So if you could do the experiment in reverse, leave the two that co-express ecdysone hormone and the receptor, leave those alive, and kill all the other ones that don't co-express the hormone and the receptor, do you think it would still be lethal?
I think so, because I can, if I release the peptide, but all the targets are dead, so what is going to do the peptide?
Right.
I think so. I know I have to test, because I'm going to do now. Now I'm in this moment of
do the combination. Okay, thank you.
Okay, thank you both. Pienso que vamos a pasar por la otra presentación porque estamos a 40 minutos. Sí. Muchas gracias, Valeria, por tu presentación. Qué bueno. Sí, y la segunda expositora de hoy es Liliana Bustos. Liliana también trabaja en el laboratorio de John. Ella es enfermera de formación y hizo sus estudios en la Escuela de Enfermería aquí en la Universidad de Valparaíso. Y luego ella trabajó en el servicio de pediatría del Hospital Carlos Van Buren de Valparaíso por 6 años. Sí. Y hasta como en 2017 ella quiso cambiar un poco el foco de su vida profesional y entró en el programa de doctorado de Neurociencia en la Universidad de Valparaíso, trabajando con John. Y hoy ella nos va a presentar algunos resultados de su tesis en una charla llamada The Drosophila Clock Regulate Ecdysone Action to Control the Daily Rhythm of Adult Emergence. Lili tiene la palabra. Puedes.
Okay. Okay, there it is. Thank you, Eugene, for the lovely presentation. Can you hear me? Yes? Okay.
Yes.
So I'm going to present some of the results that I have obtained during my PhD thesis work. So first of all, we have to talk a little bit about circadian rhythms. So circadian rhythms function to organize behavior and also physiology to anticipate and to adapt to the environmental changes that surround us, from which the most obvious is the day-to-night transitions. And in this cartoon, you can see all the physiological changes that precede our different behaviors throughout a day, like changes in body temperature and also secretion of hormones like melatonin, which happens in response to the period which should be lights off or night, and also are involved in the regulation of the sleep behavior. So these rhythms are present in a wide range of organisms, from cyanobacteria to invertebrates like insects, and also in more complex organisms like mammals and also humans. But of course, one of the most useful organisms for this for the study of circadian rhythmicity has been Drosophila melanogaster. So in the fly, circadian clock is composed by a central clock located in the central brain, and also for peripheral pacemakers located in different organs and tissues. These two kinds of clock must act together in concert to produce daily rhythmicity and also rhythmic behaviors. So in insects, one of the first behavior that is controlled by the circadian clock is also one of the first adult behavior for insects, which is the emergence from their pupa case or eclosion. And in here you can see the progression of the emergence behavior for the monarch butterfly. And just like Valeria said, we use this picture because it's a really beautiful insect, and it's similar to Drosophila. So again, back to Drosophila, if we could be able to see a developing population of flies that has been exposed to light dark cycles, we could be able to see that the emergence is restricted to dawn and the early morning. And then you can see in this graphic, which shows the pattern of daily emergence from a population of flies, plotted as the number of flies that emerge over time. You can see that emergence is restricted, peaks of emergence happens during the light part of the cycle or during the subjective day. And also during the night, flights do not emerge. Moreover, this behavior is restricted, remains under constant condition of constant darkness, for instance, and also is temperature compensated, which indicates that this kind of behavior is endogenous and not light driven. So to further demonstrate that our data sets are rhythmic, we could be able to carry on some autocorrelation analysis to show that can show us how strong our rhythms are and also how periodic they are. So you can see here that for this data set, our rhythm is really strong because in this part, it is indicated the rhythmicity index, which with values above this 0.1 cutoff show a strongly rhythmic data, and below this number, the rhythm is considered arrhythmic. And for other part, and you can see that this data is also periodic, since it possesses circadian periodicity, since it's circa a day, which means that it's almost 24 hours, almost near to that period of time. So numerous studies have shown us that the rhythm of the emergence depends on two clocks. One that is located in the central brain, and another clock that is located in an endocrine gland, which is called prothoracic gland, an endocrine gland whose only known function is the production of the molting hormone ecdysone. The lack of the central brain or the lack of the prothoracic gland renders arrhythmic the pattern of emergence, as you can see here in these plots, further confirmed by the rhythmicity index. And despite we know that these two clocks are necessary for the maintenance for the rhythm of emergence, we still do not understand how these two clocks work together to produce rhythmicity. But what we do know is that during the final molt, the brain clock neurons send time information to this other set of neurons, which are called PTTH neuron, because they produce PTTH neuropeptide, which is one of the key regulators of ecdysone synthesis. So during the final molt, time information travels to the prothoracic gland via this PTTH neuropeptide, which binds to its receptor, which is torso in the prothoracic gland, and finally produce ecdysteroid synthesis. So how can ecdysone direct all developmental transitions? Well, during the insect development, pulses of ecdysone initiate all developmental transitions, and you can see here in this graph which represent the ecdysteroid concentrations during the different developmental stages for Drosophila at 25 degrees Celsius. You can see that the levels of ecdysone must increase to cause the molt and must drop to signal the end of this of each developmental stage. So during the final molt, the levels of ecdysone increase into a really high levels and then must drop to below certain threshold to produce eclosion. So maybe since the levels of the hormone must drop, one possible mechanism for the clock to control emergence could be to work at control the levels of the hormone. However, we still do not understand how this continuous drop of the hormone can produce this discrete behavior. So our main experimental question is, how can ecdysone be involved in the regulation of circadian rhythmicity of eclosion? So one alternative could be that the clock may control the levels of the hormone. So first we want to explore if the levels of the hormone are central to the gating behavior. So in Drosophila, since in other insect species, the rhythm of emergence is achieved through the circadian control of the levels of the hormone. So here you are seeing a plot which shows us the ecdysteroid concentration measured during the final 120 hours prior to emergence. And we hypothesize that if we are able to increase the levels of ecdysone after this dramatic drop, we could be able also to move the time of emergence into a later time, but in a continuous fashion. So what we did was to inject flies with different doses of ecdysone and also the corresponding vehicle or controls, which was ethanol in this case. And during 4 times along a day, during the 4 different times during a day, which is shown here in this histogram as CT2, CT0, and so on. And then we count the number of flies that emerge over time in a period of 2 days under constant darkness. So you can see here that for these 2 controls, emergence happens mostly during the light part of the day 1. And then a forbidden area of emergence appear, which is represented here by this shaded area. And then emergence happen again. So when we inject ecdysone, what we found out was that emergence was delayed. So now flies are emerging mostly during the light part of the day 2. But interesting, we were not able to disrupt this forbidden zone of emergence. So emergence do not happen in a continuous fashion, it's still gated, which suggests that the clock is not acting at the level of the production of the hormone. So maybe it's acting downstream. So in this regard, what we did was to analyze another possibility that the clock maybe is controlling the receptor of ecdysone. So ecdysone-induced transcriptional response are mediated by its receptor, which is an heterodimer comprised by the ecdysone receptor and ultraspiracle. This dimer is stabilized by the molting hormone and recognizes specific promoter elements in the genome to regulate transcription in target tissues. Both of these proteins belong to the superfamily of nuclear receptor that mediate transcriptional response for steroid hormones. So to find out what is the role of the receptor of ecdysone, or what is the importance of this receptor in the rhythmicity of emergence, we express a dominant negative form of the receptor of ecdysone in the prothoracic gland using genetic available tools such as GAL4 UAS system, which Valeria explained it very nicely, so I'm not going to go beyond that on that. And what we found out was that with this manipulation, we eliminate the pattern of daily emergence, of rhythmic emergence, as you can see here in these traces, representative traces, and also further confirmed by the rhythmicity index, which is negative in this case, which means it is really below the 0.1 cutoff. By contrast, the record of the corresponding control shows normal circadian rhythmicity. And in here you can see the average of the rhythmicity index for all the experiment that was carried on. And black dots are representing the experimental part, and the white dots and open dots are represented the controls. Every dot represents a different experiment, and different letters represent different statistical groups. So you can see that they are significantly different. So our next step was to assess further what is the role of this receptor in the rhythm of eclosion. So now we want to express this negative form of the receptor in clocked neurons. So the first set of clocked neurons that we choose was all the neurons that express timeless. And timeless is one of the components of the central clock, of the circadian machinery. But in this manipulation, we exclude the prothoracic gland because this protein is also expressed there. So when we did this manipulation, we found out that the pattern of daily emergence was also eliminated, as you can see here in these traces, and also further confirmed by the rhythmicity index. So, but nevertheless, this neuron that express timeless and also the glia are too many. So these results could be due to severe anatomical defect in these flies, because this is expressed during all of the stages of metamorphosis of Drosophila. So then what we did was to restrict the expression of this dominant form of the receptor of ecdysone only to those neurons that express PDF neuropeptide, which is the principal neuropeptide produced by the central clock. And what we did is, what we found out was that we also eliminate the pattern of daily emergence. But despite the suspicious observation that this that the Edison signal could be necessary in the brain clog neurons for maintenance of the circadian rhythm of emergence, this arrhythmicity could still be caused by anatomical and connectivity problems, since this network of neurons needs Edison signal for proper metamorphic changes. So to explore this possibility that this could arise for anatomical deficiencies, we were to assess another circadian output, such as locomotor activity. So in here you are seeing an actogram in which every tick shows activity, and in this part you can see the days in which the experiment was carried on. And down here you are seeing the time in which is double plotted. So this part of here is the same part of here. It's just to show clearly the activity pattern of every flag. So when we express the dominant form of the receptor of Edison in all timeless neurons, what we saw was that the rhythm was pretty strong, which means that is way above 0.1 cutoff. But interesting, these flies present a significant lengthening in their period for the activity. By contrast, when we express the dominant form of the receptor only in PDF neurons, the rhythmicity index was not affected, neither was the period. So here you're seeing the corresponding control for these traces, and you can see that the rhythm was pretty strong and also pretty periodic. And you are seeing here in the right the average for the periodicity of these flies, but now in here every dot represents a different flight tested, and open dots are the corresponding controls. So you are seeing here that for this first group of flies, which are timeless flies, the lengthening in the period is pretty significant. So the next step in this assessment was to go to see the anatomy of the flies, and for this we choose this second group of flies, which was the flies that express the dominant form of the receptor only in the PDF neurons. So what we found out, in here you are seeing an adult fly brain that is expressing RFP only in PDF neurons. So this is the pattern of expression of PDF of RFP in PDF neurons. So when we express the dominant form of the receptor of Edison in this cluster of neurons, this dorsal projection branch, which is shown here by this arrowhead, and also the axon of the optic tract lobe of this other set of neurons is altered, in contrast as what can be seen in the control condition down here. So these results could reflect that, similar to previous findings in this other study in which the dorsal projection branch was shrunk, that the correct anatomical projection of this projection is not needed for the maintenance of the locomotor activity rhythms, but could be required for other circadian outputs such as emergence. So another precedent for the circadian control of the receptor of Edison came from the kissing bug, which is Rhodnius prolixus. And in here, some studies have shown that the Edison receptor is present in the nucleus during the dark phase of a cycle, and during the light part of the cycle, the receptor is present in the cytoplasm, in the prothoracic gland, as well as in other tissues. So this shuttling remains in constant condition of darkness, which shows that this shuttling is endogenous driven, but there are no evidence of this shuttling in Drosophila. So we want to see if this control was also present in the fly. And what we did was to use immunohistochemistry to assess the location of the receptor of Edison using wild-type flies under light-dark conditions, and also in wild-type flies under constant condition of darkness, to assess if if some changes that were observed are endogenously driven. So here on the left, you're seeing the pattern of expression of the immunoreaction against the all the exon receptor isoforms present in the prothoracic gland. And in here you're seeing the DAPI staining, and in the center is the merge image, which shows that the expression is nuclear. And our next step was to quantify the intensity of this immunoreaction during different times along the day. So we quantified this, and what we saw was that the intensity varies during the course of the day, being higher during the moment of the lights on period and being lower during the lights off period. Moreover, these changes remain under constant condition of darkness, which indicates or suggests that these changes are endogenously produced. But to further confirm that these changes could be driven by the circadian clock, we include a third group of flies, which are per0 mutant flies that do not possess a functional clock. And we did the same experiment, and we quantify also the intensity for the immunoreaction. And what we saw was that the intensity was dampened, but also that there is still a tiny rhythmicity in the data. So this is suggesting that for these flies, this result could be light driven and not endogenously produced. So we have to test this. We have to repeat this experiment in flies that under constant condition of darkness for per0 mutant flies. So our next step in this involvement, we wanted to see if the mRNA for the exon receptor also could be under circadian control. And for this, we used a fluorescent in situ hybridization. So we perform FISH in wild-type flies under light dark cycles. And also we include flies without a functional clock. And in here we include a third group of flies that bear a knockdown for torso, that if you remember, torso is the receptor of PTTH, and this manipulation disconnect the central clock with the prothoracic gland clock, so render arrhythmic the pattern of emergence. So essentially these two groups of flies are arrhythmic flies. And here you can see the representative images of the mRNA expression for the exon receptor at CT12, which is the point that in our cycle the lights go off. So you can see that the intensity of the expression of the mRNA is higher, way higher than for wild-type flies. So next, we classify the intensity of the expression pattern for the mRNA into three categories. So we use low, medium, and high, which is color coded in this graphic. And next, we apply a statistical test because we ask ourselves if the changes in the intensity of the mRNA expression for the exon receptor is related with the hour of the day. So what we saw was that this was only true for wild-type flies, which is what we expected. So currently I am conducting the same experiment, but now under constant condition of darkness, to assess if these changes in the mRNA expression are endogenous or light-driven. So despite all of the evidence that I present here, still another way for the clock to control the rhythm of emergence could be to act downstream of the exon receptor. So exon receptor is a transcription factor that activates a regulatory hierarchy of gene expression in which the first genes to be expressed are also called the early genes, and these early genes are also transcription factors. Interesting, some of the product of this early gene also interact with the circadian clock. For example, the product of the E23 gene it has been shown to affect circadian oscillations. So when this investigator did a knockdown for the product of the gene, they eliminate the pattern of rhythmic locomotor activity, as you can see here in this actor. Nevertheless, there is no evidence for this, that there are other effects of this product in other circadian output, like eclosion or emergence. So what I did was to conduct a bioinformatic screening using previously published list of early genes from genome-wide screening. And I select those early genes that were also transcription factors. So as you can see here, I identify a lot of genes. It's not my intention to read this or anything like that, just to show you that there were a lot of genes. And then I use these genes to conduct a second screening in which I downregulate the levels of all of these genes in the prothoracic gland to assess what happened with the rhythm of emergence when we downregulate their levels. So my results show that, as expected, the knockdown of genes that are related with the functioning of the circadian clock, such as BRIL or APE75, eliminates the pattern of daily emergence. But in addition, other genes that are related with the metamorphosis process, such as Broad or Star or Serpented leg, also eliminate the pattern of daily emergence. Thus, my results are showing that these genes are necessary for the rhythm of emergence, but also, and still, this arrhythmicity could be due to anatomical disruptions caused by the interference of exon signal during metamorphosis. Unfortunately, this is one of the best tools that we actually have for assess the involvement of the genes in the rhythm of emergence. So I hope in the future we could make some improvements and find out which is the real involvement of these genes in this process. So in summary, and as a take-home message, the levels of exon are not central for the gating mechanism or for the rhythm of emergence, but the exon receptor and downstream genes could be involved in the regulation of the rhythm of emergence. So my results suggest that the circadian clock imposes a daily rhythm to the pattern of emergence by regulating exon action in the prothoracic gland. Here are some of the publications that I have completed during my PhD work, and I want to thank to the Fly Lab, and also I want to thank to Kate, to Eugene, to Fenya, and Jean-Paul for helping me in some of the analysis of my results. Thank you.
Okay, thank you, Liliana. If they have any question for the public. Yes, Angelina. Yes.
No, estaba felicitando. Le doy la palabra a otra persona. Ya hemos discutido harto su trabajo. Felicitaciones, Lili. Lindo trabajo.
Hay otras, Kate, sí.
As your committee member, I have to ask a question. No, I'm just teasing. It was a very nice presentation, Lily. Super bien. I have kind of a theoretical question. If you look at the presentation of Valeria with the split construct used, and you look at some of that really cool slide she had with like 9 different photos of what are the outputs of the split that may interact with the exon receptor binding site, right? Do you think that some of those targets she identified could be the targets of circadian rhythm control that may be exon hormone receptor dependent? Does that make sense?
It's a closure hormone, but
I keep saying the long one. I'm sorry.
Yeah, but I think that I don't know if precisely some of this target or some of the target of ETH also receptor could be involved, because we still do not understand how this signal come out to from the prothoracic gland to the central brain again to produce all the movements that have to happen in order for the flight to emerge. So maybe one of the targets could be some of the targets that Valeria shows, but we have to make some, we conducted a bioinformatics screening showing that maybe this receptor could have some element in which exon could be bind to trigger some process that we still don't know. But bioinformatically, we were not able to find any element in the genome for the binding of exon, any exon related response element. But maybe we did, but this was just theoretically. Maybe we could look some of these targets that Valeria is showing to knock down them in some part and see what happened with the rhythm of emergence.
But in that, could you remind me in that the famous, I think it's based on Manduka, but it's also in Drosophila, the peaks of the ecdysone hormone and the eclosion hormone, there's a point at which they overlap, right? The rise of one and the fall of the other, or am I misremembering those curves?
Yes, but what happened is that, and I'm not sure if I'm going to be wrong, but in Manduka, this metamorphosis process is longer than for Drosophila.
Right.
Because in Drosophila, this only lasts 120 hours or less if we increase temperature. So maybe could be some difference at that point because of the duration of the period of metamorphosis itself. But I don't discard some of these targets that Valeria is showing.
Mm-hmm. Thank you.
Thank you, Kate. Thank you. We have a question from Maria Fernanda Palominos. He said, hormone rather than ecdysone, maybe also related one, metabolic pathway or with similar physical chemical characteristic, or it's 100% specific. I didn't quite understand what she wanted to say. That
you can read in the
but
text.
I have read the question in the chat.
Okay.
If exon receptor can be activated by other hormones rather than exon, I don't think so, because there are specific elements in the receptor of this hormone that activates them. Now what can happen is that since this is a dimer, you can form different dimers with other receptors, and maybe that could change. But in the case of the exon receptor, I don't think you can see some response of the exon receptor from other hormones. Maybe for other elements they are similar, but not other hormones. It has to be a steroid only.
Okay. Gracias. Thank you, Lily. I just have like one question about your fluorescence in situ hybridization, where you show like the wild type, the period method, and in the graph behind, I saw that in the period you have a high level of exon receptor like expression, but in the fluorescence in situ hybridization we didn't see that amount like in the photo. I don't know, maybe I missed something in the explanation, but can you maybe explain us what does mean?
I'm not sure if I understand, but you are asking me about the background. No, I'm sorry, I don't understand.
No, in the like the table you show, you have like high level of expression of the ecdysone receptor, but when you compare it to the wild type, in the graph you have like for period method flies, you have like high level, but we cannot see it like in the photos. So they are like a coin.
Yeah. Okay. That's because period mutants are highly variably. They have highly variably expression among them because they don't have like this clock that orders the expression of some elements, and one of these is the receptor of Edison. So if as we are seeing that the receptor of Edison is under circadian control, so this could explain that why you can see this variability in the data, and the picture that I show is not, it doesn't have such a great expression in comparison with wild types, because they are pretty variable. They have everything that you can see in the day. But if you look the data for wild type flies, they are pretty ordered. So the expression is increasing among the hours, and almost all the animals have a very low variability. So that's why you are seeing that you have some of the animals that have high expression, but others that have less, and others that have really poorly expression. So they have a mix.
It's very like random. Okay, thank you, Lily. Thank you, Valeria. You shared two great talks. So I don't think we have other questions. So we actualmente son las 1:13 y les digo a todos gracias por estar aquí y gracias por las preguntas y los comentarios. Y nos vemos el próximo viernes. No el próximo viernes, el viernes después, porque está el 18 que viene.